A hemopoiesis and immunogenesis system. It produces former elements of blood like erythrocytes, leukocytes, platelets, and cells participating in immune reactions, T and B lymphocytes, monocytes, and effector cells, macrophages, and plasma cells. This system consists of bone marrow and thymus, central organs, lymph nodes and spleen, peripheral organs, and mucosa-associated lymphoid tissue. Red bone marrow is a central organ of hemopoiesis and immunogenesis. It located in the cavities of spongy bone, in epiphysis of long bones, in flat bones, in spongy bones. Red bone marrow is parenchymal organ. It consists of stroma and parenchyma. Stroma are presented by reticular tissue, which consists of reticular cells connected by processes, and reticular fibers, connective tissue with special properties. Endosteum, its lining cavities of spongy bone. And their properties is quite similar to periosteum, which cover the bone from outside. Adipose tissue consists of adipose cells. They look empty because fat diluted when the histological preparations prepared. It diluted in alcohols and xylans or other organic solvents. Stroma provide the environment for development of parenchyma. It's developing former elements of blood. Stroma also include blood vessels like arteries, the branches of arteries feeding the bone, branches for capillaries and especially for sinusoid blood capillaries, very large, more than 30 microns in diameter, filling this blood contain mature former elements. The wall consists of endothelial cells with many fenesters and intercellular clefts, and basal membranes sometimes absent, so the mature former elements can go through the capillary wall and transported to the periphery. Parenchyma occupy all other places in red bone marrow. It red because there are a lot of erythrocytes. They provide a red color. In newborns or young children, all bone marrow is red. During the life to 18 years old, in diaphysis, red bone marrow replaced for yellow bone marrow, where only adipose tissue and some reticular tissue still exist. So it looks like adipose tissue in the whole. If some blood lost take place, yellow bone marrow convert to red bone marrow for some times. So parenchyma developing former elements of blood, former elements at various stages of formation and maturation. They included all these developing former elements of the scheme of Hemopoiesis. I remind you the 6 stages of Hemopoiesis. First is pluripotent stem cells. Second stage is semi-potent semi-stem cells. One is precursor of myelopoiesis and another precursor of lymphopoiesis. At the third stage, these 2 cells differentiated to 8 unipotent cells. On the 4th stage, they converted to corresponding blasts, monoblasts, 3 myeloblasts, precursors of neutrophil leukocytes, eosinophils leukocytes, and basophil leukocytes. Megakaryoblast, precursor of platelets. Erythroblasts, precursor of erythrocytes. And 2 types of lymphoblasts, T and B lymphoblasts, precursor of T and B lymphocytes. On the 5th stage, blasts differentiated to mature cells. Monocytes, 3 types of granulocytes, segmented granulocytes. Platelets, erythrocytes, and TB lymphocytes. And 6th stage, mature former elements. Only these former elements can enter the peripheral bloods. Only they can go through the sinusoid blood capillary wall. Others cannot pass. All these former elements, for exception of T lymphocytes, are developing in red bone marrow. T lymphocytes are developing in Thymus from precursors which appeared in red bone marrow. This is red bone marrow smear. If take the droplet of parenchyma and prepare the red bone marrow smear on the glass, dry, fix, color, and study under high magnification of the microscope, you can see both mature former elements like erythrocytes or lymphocytes or step neutrophil leukocyte, and also numerous undifferentiated cells. It differs the red bone marrow smears from peripheral blood smears. A red bone marrow section provides the real picture, real structure and organization of red bone marrow. You see the endosteum, the adipose cells, and even at low magnification you can see the megakaryocytes with numerous nuclei, the blood capillaries, former elements at different stages of hemopoiesis. They organized in colonies. Near the blood capillaries you can find the colony of developing erythrocytes and platelets. Far from blood capillary you can find the colony of granulocytes or B lymphocytes. In the center of colony located the colony forming units, unipotent cell, and differentiated cells located around and in the periphery, and only mature cells can go through the blood capillary wall and enter the blood circulation. Immature cannot pass this barrier. This picture demonstrates the special macrophage nurse cells with surrounding erythroblasts. Nurse cells located usually in the center of the colony of developing erythrocytes. Its process goes through the capillary wall and get iron, and feeding by iron the erythroblasts. It's necessary to the hemoglobin synthesis in developing erythrocytes. Thymus, also the central organ of immunogenesis. It provides antigen independent proliferation and differentiation of T-lymphocytes. This organ located behind the sternum. The maximal size reached to 10 years old and weight 30-40 grams. Later it became smaller and replaced by adipose tissue. Its parenchymal organ is covered by capsule and connective tissue septa, which divide in organ for lobules. It's stroma. And another stroma inside lobules composed by epithelial reticular tissue. Every lobule contains the peripheral darker part cortex and central lighter part medulla. This is the part of thymus, connective tissue capsule and tubercles and lobules. These cortex and medulla, these points are T lymphocytes. They compose parenchyma of thymus. These are epithelial reticular cells. They provide the conditions, environment for development T lymphocytes. When they became aged, they concentrated in medulla and produce lamellar bodies, Hassal's thymus corpuscles. In the center you can find the places of degenerations like keratin in epidermis. It's a distinctive feature of thymus. Some epithelial reticular cells called nurse cells. They have long processes or folds, and in these folds up to 100 T lymphocytes located, and these nurse cells provide the environment for development T lymphocytes. They teach them and examine their knowledge. How teaching of T-lymphocytes in thymus take place? First, these epithelial cells represent developing T-lymphocytes receptors for foreign antigens and check the ability to forming TCR receptors specific for foreign antigens. If they cannot produce these receptors, these epithelial cells initiate the process of apoptosis in these T lymphocytes and they died. T lymphocytes which were able to pass this exam move to the next region where another epithelial cells present them the antigen of their own body, their own antigens, and check if they able to produce specific receptors. If they do it, they initiate the apoptosis. It called positive and negative normal involution. When this age, Thymus parenchyma replaced by adipose tissue. And accidental involution. Accidental is mean any severe stress or disease when stress hormone glucocorticoids of adrenal cortex release into the blood and stimulate the death of T lymphocytes, especially in thymus cortex. As a result, the cortex became like medulla, no differences between cortex and medulla inside lobules of thymus. But when the stress or disease is over, the structure of thymus can return to the normal. Lymph nodes are the peripheral organs of hemopoiesis in immunogenesis. Antigen-dependent proliferation and differentiation of B and T lymphocytes take place here. There are several hundreds of lymph nodes in the body. They are located on the way of lymphatic vessels. Their size between 5 and 10 millimeters. They're bean in shape. Of its convex aspect, several lymph vessels enter the node. On the concavity, hilus, one vessel leaves the node. They are afferent and efferent vessels. Lymph nodes are parenchymal organs. They consist of stroma, connective tissue capsules, and thin tubercles. Inside, the reticular tissue also composes additional stroma of lymph node. Parenchyma represented by T and B lymphocytes. At the periphery, they compose the lymphoid follicles. They situate in line in one plane and produce the cortex. And medullary cords consist of lymphocytes located in medulla, the lighter part of lymph node. You can see here at this picture the reticular fibers, part of reticular tissue, after the special silver impregnation. You can see also how the artery enter the lymph node and how veins leave it. And you can see also how the lymph flow through the lymph node, through the lymphatic sinuses. Here is a picture of the piece of lymph node: capsule, connective tissue tubercles, lymphoid follicles consist of B lymphocytes. They concentrated on the periphery of these follicles. In the center, the lighter part, germinal or erective center. Here the lymphoblasts proliferated. Medullary cords. Follicles and medullary cords are B-dependent zones. They composed of B lymphocytes, and there is T-dependent zone between, paracortical zone, composed of T lymphocytes. Consequently, here and here, B lymphocytes, antigen dependent proliferation and differentiation take place, and here T lymphocytes contact with antigens and proliferate and differentiating. So the lymph flow through the lymphoid sinuses. First subcapsular, then cortical, then medulla, central sinus, and they leave the lymph node. Lymphoid sinuses, lymphatic sinuses, lining by endothelial cells, like all lymphatic capillaries. But among the endothelial cells there are the special macrophages which can phagocyte foreign particles, bacteria, and purify the lymph. And you can see them if you inject into the lymph, for example, the blue dye. Macrophages of sinuses uptake the dye from lymph and stain themselves in blue. B lymphocytes provide two types of effector cells: plasma cells, which produce hemoglobulins antibodies, which circulate, release into the blood, and response for humoral immunity, and B memory cells. And T lymphocytes produce T killers, T helpers, T suppressors, and T cells of memory. This picture demonstrates the circulation of lymphocytes. You see, they enter the lymph node, pass through the lymphatic sinuses, and then flow through bigger and bigger lymphatic vessels, reach the veins, with venous blood go through the heart, then arteries, and they can come back into the same lymph node and go out through the wall of postcapillary venules covered by prismatic epithelium, special endothelial cells. Spleen. It's also a peripheral organ of hemopoiesis and immunogenesis. Its typical parenchymal organ consists of stroma and parenchyma. Stroma represented by thick capsule and tubercles. They contain many smooth muscle cells, and they can contract and push the blood spleen vein, and they can relax and accumulate a lot of blood, up to 1 liter of blood. Inside spleen there is additional stroma, reticular tissue, like in red bone marrow and lymph nodes, which consist of reticular cells connected by their processes and numerous reticular fibers which produce the network for conditions for parenchyma. And parenchyma represented called the pulp, and there are two types of pulp: white pulp and red pulp. White pulp consists of lymphatic follicles, quite similar to lymphoid follicles of lymph node, but located irregularly, and every contain the central artery at the periphery. Red pulp are located between follicles and tubercles. At high magnification you see the capsule, thick capsule and tubercles, and capsule covered by mesothelium, the visceral peritoneum. You see the reticular tissue inside, reticular cells. These are lymphoid follicles which compose the white pulp, and between there is a red pulp. Red because a lot of erythrocytes in sinusoid blood capillaries and inside stroma. The erythrocytes come here for death. The aged, old erythrocytes come here and died and phagocytes by macrophages. It's serve like the cemetery for erythrocytes and platelets. White pulp composed by T and B lymphocytes, and there are 3 zones. Proliferation center or germinal center, it's B zone. Here B lymphocytes proliferated. Periarterial zone belong to T lymphocytes, and mantile, this dark zone. And marginal zones contains a mixture of T and B lymphocytes. This slide demonstrates blood circulation in the spleen. Blood enters the spleen through the spleen artery. This branching for trabecular artery, pulpar artery. When it goes through the lymphoid follicle, it called central artery. Then it branching for arterioles, penicular arterioles. They continue into capillaries, which covered by chaethes. They work like sphincter and regulate the entrance of blood into sinusoid blood capillaries, or release blood into the stroma. So this is close, normal, usual, close circulation, venous sinuses, pulpar veins, trabecular veins, and spleen vein, and open circulation when blood leave the blood vessels and enter the stroma, and then collected by sinusoid blood capillaries. This is schematic drawing of open and closed blood circulation in red pulp. This is a sinusoid blood capillary or venous sinuses. It linked by long endothelial cells with intercellular clefts between and no basal membrane, so the circular spiral special reticular fibers support these endothelial cells. And when the blood flow through these capillaries, it's closed circulation, old aged erythrocytes leave capillaries through the intercellular cleft of endothelium and phagocytate destroyed by numerous macrophages. They destroyed hemoglobin and iron and the blood stream, and uptake by special transport proteins, transferrin, and transported to the red bone marrow, uptaken by special macrophages, nurse cells, and which convey the iron to development erythrocytes for hemoglobin biosynthesis. And open circulation when blood leave the blood capillary and macrophages destroyed aged old erythrocytes, and young normal erythrocytes return to the bloodstream through the clefts between endothelial cells. On the histochemical preparation you can see pink white pulp, no macrophages, no iron here, but in red pulp in stroma there are numerous macrophages around sinusoid blood capillaries which accumulate iron, and this iron stain in blue. This is scan electron microphotogram of red pulp, sinusoid blood capillary wall, endothelial cells with large intercellular clefts, reticular cells of stroma, and numerous macrophages which waiting for old erythrocytes and destroyed them. Mucosa-associated lymphoid tissue consists of numerous lymphoid follicles distributed almost everywhere in the body, especially in mucous membrane of alimentary tract and airways of respiratory system, urinary passing organs of the urinary system. In digestive tract, this follicle concentrated inside tonsils. You know, there is 5 types of tonsils between oral cavity and pharynx. Do you see the folds of mucous membrane, and under the epithelium there are numerous lymphoid follicles like in lymph nodes. And in the gut, in small intestine, you can find the single lymphoid follicles and aggregated lymphoid follicles. Their patches consist of several hundreds of lymphoid follicles. And in large intestine there is appendix vermiformis. In mucous membrane of it there are also numerous lymphoid follicles.